All attendees are in listen-only mode.
Welcome everybody to today's webinar presented by Biognosis, talking about fast proteome quantification using Micro Flow SWATH. Today with me joining for this webinar will be Nick Morris from SciEx.
Hello everyone.
The outline of today's webinar is that I'll present first a short introduction into the samples, this study setup, what kind of spectral libraries we used, and kind of the overall strategy. Nick will then introduce the SWATH technology, talk about the microflow setup, give a little bit more details about the methods that we have used on the TOF 6600s, and then I will take over again to talk about the data analysis done in Spectronaut, looking at how we can actually derive biological meaning out of the proteomics data. And in the end, as always in our webinars, we will have a short question and answering session where Nick and I will be happy to take any questions that come up during the webinar. So please hold your questions up until the end. And as always, also, the webinar will be recorded and a link for the recorded webinar will be sent to all registered people after the webinar. In this study that we present today, we actually worked on biopsies from 3 different patients suffering from liver cancer. So what we obtained was 3 liver biopsies from healthy tissue and adjacent cancer tissue, which we have processed in-house using the Biognosis Sample Preparation Kit Pro. And we then sent the digested and cleaned peptides to SciEx, to Nick, so that he can acquire both the DDA for spectral library, but also the SWATH data on the TOF 6600 for the actual quantitative work. On top of the spectral library produced directly from the sample, so the project-specific spectral library, we have also used 2 resource spectral libraries, one the pan-human library and the other one the resource liver library that is also that you can actually download within Spectronaut. And we've basically used these 2 bigger libraries to show that nowadays with the protein FDR calculation on the actual DIA data, you can actually also use these kind of bigger, larger resource spectral libraries to generate meaningful data. I'll now hand over the word to Nick so that he can actually introduce the SWATH workflow and the TOF.
Thanks very much. As most of you may know, SWATH is a data-independent method whereby we acquire data across mass windows, normally from M/Z 400 to 1250 on the TOF. And basically all precursors are fragmented, and then we match the MSMS data to spectral libraries that Florian was outlining in the previous slide. Just checking if I have control here. Could you advance the slide, Florian? Sorry, there we go. So we have these SWATH acquisition windows where we step across the mass range and we acquire relatively high resolution MSMS spectra in a chromatographic time frame. Sort of resolution that the TOF will give you will be around 25 to 30,000 resolution. And this generates targeted data that gives us high quality quantification data. Just advance the next slide. So this animation here basically shows how we step across the molecular weight range, and of course the MSMS spectra that's acquired is not from individual precursors, but they are composites of many precursors. And we quantify at the MS2 level rather than MS1, using definitely Spectronaut here with Biognosis. And we also use our own software package as well. Both are complementary. So the Q1 isolation strategy is that we would step across the molecular weight range, the M/Z range, and acquire the MSMS spectra. And we normally try and do this in the shortest possible time. We have found over the last few years that rather than doing fixed window swath, which was proposed very early on, we now use variable window swath, because with the narrower windows with the highest M over Z density, we will get higher quality MSMS spectra with less and less spectral interference from co-isolated deuterites. So this graphic on the right here shows a normalized density of precursors across the whole LCMS run, and how where the most abundant, the highest density of precursors here shown around M over Z 600. This is the region where we have the smallest isolation windows down to 4 Daltons. And in areas where there are fewer precursors, i.e. greater than M over Z 1000, we will use much wider windows, normally up to about 25 Daltons. The whole outline of this presentation is about microflow swath, and this is maybe somewhat of a new technique for some people. It's a very, very reliable and very robust technique, whereby we're using 300 micron ID columns at 5 microliters a minute. And the beauty of this is it's very, very easy to set up. Column equilibration time is very short, normally just a couple of minutes, and the retention time reproducibility run to run can be incredibly good. It's usually less than 10 seconds for any precursor. The gradient that was used for the swath methods we're going to outline here is shown on the right. So it was a total runtime of 57 minutes. And the actual gradient part was 45 minutes. And so the run-to-run injection time is not nominally 1 hour. So you can usually process up to 24 samples in one day. The setup that, with the triplet off is that we use the dual spray source, which has been fitted with a dedicated microelectrode, which is 50 micron internal diameter, which is, you can see on the top of the source here. And it's just connected to the column using a short length of 50 micron peak seal. The total duty cycle that we use for our swath method is only 2.3 seconds. And so normally we're looking for peak widths of about 20 seconds at base or thereabouts to give us 8 points across the peak for quantitation. The swath method itself is set up using a calculator, and this 100 variable window swath method that we describe here is actually a default method used, for example, in Skyline. And now I should hand back to Florian.
Good. Thanks, Nick, for the introduction about the microflow setup and the triple TOF and how we actually set up the method. I'm now going to talk about, so once we got the data back from Nick and his team acquired on the triple TOF 6600, we first of all used the Protein Pilot 5.0 output from the DDA runs to generate a spectral library within Spectronaut, and we controlled the spectral library quality at a 1% global FDR. The spectral library created with this shotgun files was in total a little bit over 22,000 precursors large, spanning 2,570 protein groups. And then, as mentioned, additionally, we have also used the pan-human library, which was published by Rudy Ebersol's lab, which you can actually download directly within Spectronaut. On top of that, we've also used a resource library that also can be downloaded directly from Spectronaut. The resource library here is a liver library that we have acquired at Biognosys in-house. For the analysis of the swath data using the three described spectral libraries, we have controlled both the precursor and protein group or protein FDR level at 1% using the latest release of Spectronaut, Spectronaut Pulsar, and Spectronaut 11. As you can see in the upper right picture, basically we could identify up to 21,000 precursors using the DDA-based library, and larger resource-specific libraries, they actually identified more precursors, both for the liver resource library and the pan-human library. The liver library kind of a little bit worked better, showing that these tissue-specific resource libraries are better used than the global libraries, such as the pan-human library. Also, in terms of protein groups here depicted as cumulative identifications, the resource library performed best, followed by the pan-human library, and then followed by the DDA library. For the quantification of the SWATH data, we then used the Q-value sparse percentile filtering, at which we set the percentile filtering to 0.5, meaning that in order to quantify a peptide precursor, it needs to be identified in at least 50% of all runs. And the underlying assumption here was that there are peptide precursors and proteins that are actually only expressed in either of the two conditions. So there are proteins that are only expressed in cancer, and there are proteins that are only expressed in healthy, but still we want to quantify these differences. Now, as you can see on the lower right picture, using this 0.5 percentile filter, the DDA-based project-specific library performs best, meaning that with these project-specific libraries, you are identifying more reproducibly the same peptides over and over again compared to the large resource libraries. We then also performed all the automated statistical tests that Spectronaut is doing. So we did a pairwise t-test to find statistically significant candidates. On top of that, we also did unsupervised clustering using now the DDA library only for further analysis. And what we could see first is that so the two conditions, cancer and healthy, they are clustered together, but also that the three technical replicates of each sample actually clusters close together. We also looked at the technical CVs of these run replicates, and what we could see is that the technical median CVs at the precursor level were below 10%, showing that the microflow setup on the TripleTOF 6600 is a highly robust setup. And you can basically omit doing technical replicates and save that time for biological replicates. Then, using the statistical test in the pairwise t-test in Spectronaut, we identified 594 proteins being significantly differentially regulated between the cancer and the healthy samples. Here, the cutoff criteria that we used was a Q value of smaller than 0.01. Here, the Q value means the P value corrected for multiple testing, and the absolute log 2 fold change needs to be bigger than 1. On top of doing the statistical analysis in Spectronaut, we also used the data to do biological interpretation using the Ingenuity pathway analysis, or also known as IPA. First of all, we looked into our candidate data set, and we found that 18 out of these 594 proteins are hepatocellular markers according to Oncomine. And we then loaded the data, as I mentioned, into IPA. And what we could see here is that several degradation pathways are being affected. And in most of these degradation pathways that are affected, the proteins that are in these pathways are downregulated in the cancer samples, meaning that the cancer samples are actually show an impaired detoxification capabilities. On top of directly analyzing the protein candidates and the quantities that you got from the proteomics measurements, IPA is also capable of looking at upstream regulators and giving probabilities about the deregulation of these upstream regulators. The most significant upstream regulator that we found in these data sets being affected is the microRNA miR122, which is a known tumor suppressor. And of course, if you're if you're actually deregulating a tumor suppressor, then the proteins that are downstream of these regulators are also either up or down regulated, as depicted here in the left image. Out of 17 proteins regulated by MIR122, we could show that 15 were upregulated in cancer as expected. For some of the proteins, like SEP9, you can actually nicely see here that it's significantly higher expressed in the cancer samples, whereas in the healthy samples it's lower expressed. For other proteins, the regulation was not that clear, and you can see that it's only for partial samples actually regulated. This also already nicely indicates that how precise you can basically monitor protein expression among different patients. Good. With this, we're already coming to the summary of today's webinar. And I hope Nick and I could show you that with a very small study, just using 6 samples, we could basically show a little proof of principle that the microflow SWATH setup is a very robust setup that can be used in high throughput to measure a lot of samples in very short time. In this case, we have measured 18 samples in less than 1 day, meaning that this study can easily be extended to larger cohorts because of the high throughput using the microflow setup. We are able to measure around 150 proteomes a week. In Spectronaut 11 and Spectronaut Pulsar, with the implementation of the protein FDR concept as recently published by Rudy Ebersol's lab, we can actually now use also resource spectral libraries such as the pan-human library or tissue-specific resource libraries that are directly implemented in Spectronaut. Still, the project-specific spectral libraries perform best in terms of reproducibility using a 0.5 Q-value sparse percentile filter. Overall, we could, in this small proof of principle study, we could show that 594 proteins are statistically significantly up or down regulated between the cancer and the healthy samples. And I hope I could also show that by using the IPA tool, you can actually draw meaningful conclusions out of the data directly, but also using the tools that show you about that give you information about the upstream regulators of these proteins. This entire study will also shortly after the webinar be published on our Science Hub as an application note, and you can read it and look more in detail into the data. With this, I'd say thank you for your attention, and Nick and me will now take questions. If you have any questions, you can actually just type them into the questions box or into the chat. Okay, so we got the first question is, what is the minimum amount of sample required for injection using the microflow setup?
That's a good question. We normally find the difference in sensitivity between nano and micro about 4 to 5-fold difference. So we routinely analyze around 4 micrograms of cell or tissue extract. You can go lower than that, you can go down to 1 microgram. The number of proteins that you will quantify as you go down will start to drop off. But it's low microgram amounts of material will give you good proteome coverage.
Good, thanks. The next question is about the challenges that you anticipate when measuring very large sample sets and how to overcome those. So in-house at Biognosis, when doing very large experiments, such as the recently showed Diogenes project, what we're using is dedicated QC samples that we're using to then do batch correction for these large-scale studies. The next question for you, Nick, again, is about the sensitivity of the microflow SWATH setup, if you can comment on that.
Well, as I said, we see about a 4 to 5-fold difference between the sensitivity between nano and micro. Certainly, when we look at peptide levels, we can certainly see sub-femtomole, high attomole levels of peptide quite clearly. It's not the dramatic loss in sensitivity that most people think when they switch from nano. So if you are used to using nano and wanted to switch to a higher throughput, you're not going to be compromised too much on overall sensitivity.
Good. The next question is about the overall quantification mentioned about the DDA library. And here, what I wanted to say is that the overall quantification means this is the cumulative number of peptide precursors and protein groups identified, meaning that every peptide precursor and every protein group that is identified at least once is counted here. And because for the quantification, we want to go into more the more reproducible peptide precursors that are identified, we use the sparse percentile filter, and then did the t-test only on that. Next question is about the DDA library. In this library, we did not do any fractionation for the DDA library, but we just simply measured the 6 different samples in shotgun mode. And of course, by applying fractionation such as HBRP, you can actually significantly increase the size of your spectral library, and thus also increase the number of peptides and proteins that you can identify. In-house, we are generally advising people to use anything between 6 to 12 fractions of the condition pools. Then the next question is about the technical replicates. As I mentioned, we have measured every sample in technical triplicates, so those technical triplicate re-injections. And for these, the median CVs of on the precursor level were below 10%. The next question for you, Nick, is about the LC, and can it be shortened to something like 30 minutes?
You can. We have done this. You do start to lower the number of proteins you will quantify, because as you make the gradient shorter, your peaks get sharper. And with the duty cycle for the swath at around 2 seconds, the number of points across the peak is actually quite important. With a half an hour run in microflow, you might have peak widths at base of maybe 10 seconds or less. So you can, but you may need to adapt your swath method.
Thanks for that. The next two questions are related. So the first is how we selected the normal tissue. So here the selection of the tissue type was done by the provider of the tissue, where we basically just ordered healthy versus cancer tissues, and it was done by the pathologist. So they actually annotated the healthy and the cancer tissue. Then, as I mentioned before, the liver tissue library that is available in Spectronaut, you can download it within Spectronaut. Just make sure that your computer where Spectronaut is running is connected to the internet, and that the firewall is not blocking the access to our server. Good. Are there any other questions considering this study? As I mentioned before, the study will also be soon after available on the Science Hub at www.biognosis.com, and then actually you can download it, read it, and if you have more questions, you can actually send them to us. Good. This, I would like to again thank you all for joining this webinar today. Also, special thanks to Nick for actually being here together with me to answer any questions and present the Microflow SWATH setup. And I wish you all a very nice day.